Well, the simplest way to put it is that scientists had an idea to make crops more resilient to external factors. So let's say you grow tomatoes, and at night it gets so cold that by morning half your tomatoes are frozen, and now you can't sell them anymore. This is what scientists were able to change through genetic modification. They figured out a way to take DNA from one organism and put it inside a completely different organism to create a desired and specific outcome. For example, let's go back to the tomato scenario. Scientists recognized that Arctic flounder, yes, that's the fish, could withstand extremely cold temperatures. They thought, what if we took the DNA trait that allows the flounder to withstand the cold and put it inside the tomato DNA, so that the tomato could withstand the same temperatures? Now you've just narrowed your chances of your tomatoes becoming frozen. This idea that you could now get 100% crop yield was the original guiding principle for genetic modification. Our friends at Lily Films have a documentary called The Future of Food by Deborah Coons-Garcia that explains it best.
Genetic engineering is really a radical revolution in food production. It's really a cell invasion technology. You know, people have heard they're taking a flounder gene and putting it in tomato so the tomato can last in cold temperatures. But people don't ask, how does that flounder gene get in that tomato? How does it get in there? And what really happens is, the only way you can do it is to invade the cell of the tomato and deposit the flounder gene. Well, what's good at invading cells? Bacteria and viruses.
After 12 years of searching, Monsanto found a soil bacteria that is naturally immune to Roundup herbicide. Their goal was to genetically engineer DNA from these bacteria into various plants. They cut out a sequence of DNA that is resistant to Roundup. But if this DNA sequence alone is inserted into a corn plant, it will have no effect. So the next step involves E. coli bacteria. Gaps are created in the E. coli DNA, and when the two test tubes are mixed together, some of the E. coli DNA recombines with the Roundup-resistant bacteria. Then the technicians smuggle the engineered DNA into the cells of the corn plant they want to modify. Cells will naturally reject foreign DNA, so they developed a method using soil bacteria that causes tumors in plants. They use this bacteria to ferry the engineered DNA into the plant's nucleus. There are also two other methods used to get the engineered DNA through the cell wall. One uses a stream of electricity to create tiny holes in the plant cells so they become vulnerable to infiltration by foreign DNA. Another is the gene gun, which blasts particles of gold coated with engineered DNA into the plant cells. Each of these three methods needs a promoter gene that turns on the desired characteristics. The promoter gene is often extracted from the cauliflower mosaic virus.
This capacity of bacteria and viruses to invade mammals in different ways is what really has a lot of people edgy about biotechnology, because that's really what the tools are all about. In order to move genetic material from one organism to another that don't normally cross, you've got to sort of behave like bacteria and viruses and invade into the cells and become established, just like a virus must become established.
American Heritage Medical Dictionary defines genetically modified organisms as an organism whose genetic characteristics have been altered by the insertion of a modified gene or a gene from another organism using the techniques of genetic engineering. The hope was that this biotechnology would end world hunger by creating higher crop yields. Ironically, we have found studies that indicate just the opposite. Reports have shown that genetically modified crops actually produce fewer yields. In fact, in some countries where starvation rates are very high, there have been many cases of citizens burning genetically modified crops in protest. Like the Future of Food said, Monsanto, the largest biotech company in the world, was able to make seeds that resisted their own Roundup herbicide. This allows farmers to spray as much herbicide as they want without worrying about their crops dying. But it doesn't stop there. Scientists can now make seeds that produce their own pesticide. This is like you and me walking around with mosquito repellent coming out of our pores. How do they do this? Scientists found a bacteria in the soil that is lethal to many species of insects called Bacillus thuringiensis, or BT for short, and they genetically engineered it into the crops. So what could the effects be when the bacteria that only used to live in our soil is now being distributed into the air through our crops? In the Philippines, the people living next to the genetically engineered cornfield developed recurring skin, respiratory, and intestinal problems. Blood tests were done on 39 of the Filipinos, and it was revealed that their immune system was affected by the BT toxin that the corn had released. And there's still more. Instead of just making drugs in labs, scientists have been growing crops that produce their pharmaceutical drugs, like research chemicals, blood proteins, antibodies, and vaccines, just to name a few. According to the Society of Diabetic Rights, thousands of diabetics have experienced bad reactions, and many have died as a result of this genetically engineered insulin called synthetic or human insulin. The Union of Concerned Scientists have been urging the United States Department of Agriculture to put a ban on pharma crops due to the potential that they could contaminate all of our food supply. Our next logical question after finding out how this all works was, is it safe? Should we be eating genetically modified food? What do you think?
Dr. Ermakova of the National Association of Genetic Security in Russia wanted to see what the effect of eating genetically modified foods would be on rats and their offspring. The experiment had 2 parts. In part 1, there was 3 groups with 3 rats in each group. Let's call them Group A, Group B, and Group C. She fed each group of rats food 2 weeks before conception. In Group A, she fed the rats no soy products. In Group B, she fed them soy flour, and in Group C, she fed them genetically engineered soy flour. Then came part 2 of the experiment. This time, there were just 2 groups of 3 rats. Let's call them Group D and Group E. In Group D, she fed the rats food with no soy products. In Group E, she fed them genetically engineered soy flour. So in total, Group A and Group D ate no soy products, and those rats gave birth to 44 babies. Group B ate regular soy flour, and those rats gave birth to 33 babies. Group C and Group E ate genetically engineered soy flour, and those rats gave birth to 45 babies. Well, that sounds normal. Well, 3 weeks later, the results were a little shocking. In the group with no soy at all, 3 baby rats died. In the groups with regular soy, 3 baby rats died. But in the groups who ate genetically engineered soy flour, 25 baby rats died. According to Dr. Ermakova, the reason why this is a problem is because the biochemical structure of rats is very similar to the biochemical structure of humans. The studies done on other animals resulted in precancerous cell growth, smaller brains, livers, and testicles, damaged immune systems, liver malfunctions, lesions in the liver, stomach, and kidneys, inflammation of certain organs, cell malfunctions, higher blood sugar levels, fertility problems, and unexplained increases in death rate.
All right, welcome to the Health Ranger Report. This is Mike Adams, the Health Ranger, reporting for naturalnews.com. Today we have some new information, breaking news from a medical journal called Cell Research. This is news about the dangers of genetically modified foods, and this is new information. This shows the mechanism by which genetically modified foods, or GM foods, or GE foods, genetically engineered, may alter your organ function. You see, in the past, Monsanto has said that genetically modified seeds and foods are presumed to be safe, presumed safe. It doesn't mean they're proven safe. In fact, right here from the Monsanto website, a couple of quotes you need to know. Here it is. Monsanto says, quote, "There is no need to test the safety of DNA introduced into genetically modified crops." And they go on to say, there is no need for or value in testing the safety of GM foods in humans. So you see, Monsanto has a position that they don't even need to test the safety of these genetically modified foods because it doesn't even matter. But now here in Cell Research, we have a new study called the exogenous plant MIR16A. That is a microRNA study that we're talking about here. Specifically targets mammalian LDL-RAP1. That's a receptor for LDL cholesterol function in the liver. Evidence of cross-kingdom regulation by microRNA. Now, it means that microRNA, which is information, it is genetic information, has now been found to pass from the foods through digestion into your blood and then to attach onto your organs. You got that? It goes from the food through digestion into your blood and attaches onto your organs, and there it modifies the function and the expression of those organs. This is groundbreaking science. This nullifies the safety claims of Monsanto. And it means that we are more than just the vitamins and minerals and fiber and carbohydrates and proteins that we eat. It means we are also eating information. You got that? We're eating information because DNA and RNA is information. So when we're eating this microRNA, it's surviving that process and then providing information to the cells and the organs in our body. Maybe this is why GMOs are causing such widespread infertility, because the microRNA is attaching to the receptor sites of the fertility organs and altering the expression or the function of those fertility organs. Let me read you some more from the study because it's quite fascinating. This is in the abstract of the study, which again was published in the September 20th edition, 2011 of Cell Research. The abstract says, quote, "Here we report the surprising findings that exogenous plant microRNAs are present in the sera, that means the blood serum, and tissues of various animals, and that these exogenous plant microRNAs are primarily acquired orally through food intake." That, again, is breaking science. It changes everything. You see, you already know you are what you eat. And normally that means you are the vitamins and the minerals and the other nutrients that you consume. We know that that's true on a physical level or even a biochemical level. Now, what happens when you consume plants that have been altered, artificially concocted in a laboratory by profit-driven scientists working for the most evil corporation in the world? You get information that's bad information. It's not natural information. It is frankenfood information, and when you consume that, that information goes into your body and then begins to program your organs and your cells and your tissues to behave in a way that is artificial, that is not natural, that is Frankensteinian in its effect. MicroRNAs have been widely shown to modulate various critical biological processes, including differentiation, that's cell specialization, apoptosis, which is part of the cancer prevention process in normal human cell metabolism, proliferation, the immune response, and the maintenance of cell and tissue identity. Now, that's another crucial statement. What do you get when your cell doesn't remember what identity it's supposed to express? You get a DNA mutation that results in the formation of a cancer cell that goes on to become a cancer tumor. So
people say, hey, Alex, don't you care about this election or that or this? No, I care about World War III happening or financial collapse, and I care about the poison water with the fluoride, the GMOs, the aspartame, the mercury, all of this stuff. And Mike Adams, the Health Ranger, has put together huge breaking news. It's at Infowars.com and naturalnews.com. It's the top story, linked in Reddit, Infowars.com right now. New GMO study, rats fed lifetime of GM corn grow tumors. 70% of females die early. This in big new studies. Now, there's been a lot of other studies as well showing within 3 generations near total sterility, 99%, hair growing on the tongues of the rats, the guinea pigs. But this is a new big one. You want to know what the New World Order stands for? It's not just about grabbing power. They're poisoning everybody. They're eugenicists. And joining us is Mike Adams. I know he's very busy today. He's getting ready to launch a lot of new stuff, so he's not been able to come on a lot lately. He will be on the show Monday, by the way, with some more breaking news. But Mike, my friend, break down this huge report with photos. And we'll be showing some of those to PrisonPlanet.tv viewers. Radio listeners, just go to naturalnews.com or Infowars.com and see it for yourself. But Mike, break down what we're facing here.
Yeah, the photos are shocking, Alex, and the study found horrifying tumors growing at a 200 to 300% increased rate in these rats that were fed a lifetime of genetically modified corn. This is Monsanto's variety of GM corn that they fed these rats. You know, you've been covering today the bullets, the DHS bullets, 1.6 billion rounds of bullets. That's a huge story. It is related to that in the sense that food is now a weapon.
Kill us, and then we creep back 10 years later dying, and they suck all our savings out of us at a Rockefeller death camp known as a hospital.
Yeah, if they don't get you with the sniper rounds of the ballistic 308 bullets that they're buying, they'll get you with the food. That's what this is all about. Now, here are some of the big findings. 70% of females suffered premature death, and up to 50% of males suffered premature death. Severe organ damage in those who ate this for a lifetime. We're talking liver damage, kidney damage, damage throughout the entire body system, all of biology damaged by this. We're talking a massive increase in large tumors. Tumors so large, Alex, that I don't know if your producers can show some of the pictures on screen. They are horrifying. It shows tumors so large that some of these rats had to drag the tumors across the floor as they were trying to crawl.